Good evening, everybody, and thank you for your presence, whether you unite with us from campus or across time zones. Tonight marks the 35th chapter of our Mentoring Talks series, a hallmark initiative of the American University of Beirut's Transformative Education TRED program. This is our first mentoring talk in 2026, the year that marks the 160th anniversary of our beloved AUB. If it is the first time you join us, know that you are crossing into an area where questions matter more than credentials, where science meets storytelling, and where the flame of discovery is collaborative, student to scientist, mentor to mind. At TRED, we fundamentally redefine academia from within, driven by the belief that the pursuit of knowledge should empower every student to transcend limits and realize their scientific aspirations. This inspiring series never aimed to be a platform for sophisticated profiles or precise responses, but to humanize science, revealing the uncertainties and the serendipities that power real breakthroughs. Our mentoring talks welcome pioneering minds which illuminate the pathways of discovery, not by following a predetermined route, but by questioning how these paths were made to begin with, offering insights into the intricate cognitive frameworks behind their achievements. This mentoring talk is organized in partnership with the prestigious Angewandte Chemie. I would like to thank Dr. Suzanne Toby, Executive Editor of Angewandte Chemie, for joining us on this panel. Tonight, we are truly honored to welcome an individual who has not only built his own path, but quite literally helped illuminate the way for countless others. Professor Martin Chalfie, a distinguished neurobiologist and Nobel Laureate who never fails to remind us that science does not originate solely in the lab or through funding, but rather in the courageous leap of curiosity and the silent bravery of perseverance. In the early 1990s, while others dismissed GFP as a mere scientific curiosity and oddity, Professor Chalfie saw possibility. What began as a casual Friday afternoon experiment, not for publication, not for a grant, but simply out of curiosity, ended up illuminating live cells. Thus, what started as a single glowing protein isolated from a jellyfish became a tool that revolutionized molecular biology and medical research. His discovery opened a new era of visualizing life itself, earning him global recognition. But Professor Chalfie's legacy goes far beyond the lab. As chair of the National Academy's Committee on Human Rights, he has defended silenced scientists across the world. He has spoken out on climate change, mentored generations of researchers, and reminded us that science is not about results or discovery. It is about courage, curiosity, and compassion. Professor Chalfie's journey from a swimmer at Harvard to a Nobel laureate is not a story of linear brilliance. It is a story of returning after struggle, of trusting wonder after convention, and of choosing growth again and again. Through his words and actions, he reminds us that greatness lies not in perfection, but in perseverance. And perhaps most importantly, Professor Chalfie's journey shows us that the most transformative breakthroughs in science don't always begin with the problem to solve, but with the question to explore out of pure curiosity. Ideas once categorized as useless knowledge with no funding, no purpose, can become the bedrock of discovery. As he once said during an interview with the Howard Hughes Medical Institute, HHMI, in 2014, quote, Most important discoveries came from people doing things for no particular reason other than they were interesting. End of quote. His story is a testament to what happens when we nurture intellectual curiosity and honor wonder. Science never starts with certainty, but with the bravery to ask. Professor Chalfie's example encourages each of us to trust our questions, to stay curious despite doubt, and to remember that sometimes the simplest act of wonder can lead to discoveries that change the world. Without further ado, please join me in welcoming 2008 Chemistry Nobel Laureate Martin Chalfie. Professor Chalfie, the floor is yours.
Thank you. I'm overwhelmed by that introduction. It was very, very nice. I appreciate the opportunity to talk with people and to talk really more generally about science and a little bit about how I think science is done. The title of my talk, as you see on the first slide, is rather unusual. It's the continuing need for useless knowledge. Now, the reason it's the continuing need is the phrase useless knowledge that I've used here is actually taken from a paper written in 1939 by this man, Abraham Flexner, who wrote in Harper's Magazine about the usefulness of useless knowledge. Now, Abraham Flexner is a very interesting personality in American educational history because he is the person in about 1910 who is in charge of a committee. He was never a doctor, but he was in charge of the committee that decided what all undergraduates in the United States needed to take in order to get into medical school. And we still follow that guideline of taking organic chemistry and physics and other general chemistry, biology, and mathematics as prerequisites to medical education. In addition to that, he was the person who came up with the idea and was the first director of what was called, or it still is called, the Princeton Institute for Advanced Study. This is where Einstein, Oppenheimer, Gödel, many illustrious mathematicians, physicists, economists, and others have worked and were encouraged. And he was the one that came up with the idea that there should be a research institute and was the director. So he has had a very profound effect on the development of ideas and discoveries in the United States. In this article in 1939, as you see, he wrote that intellectual life on the surface is simply a useless form of activity, but it is unexpectedly the source from which undreamed of utility is derived. And he starts the article with a very interesting story. He talks about talking to this man, George Eastman, and asking him. George Eastman developed the Eastman Kodak Company, was a very prominent industrialist, and Flexner says that he asked George Eastman, who is the most important scientist in 1939? And Eastman says without hesitation, it's of course Marconi, because Marconi came up with radio and wireless, and these are very important discoveries. And Marconi actually won a Nobel Prize for this as well. And interestingly, Flexner says, no, he's not. He is inevitable. That the real basis of radio and wireless comes from an understanding of electromagnetic waves and their properties. And this is the result of the research, not by Marconi, but by James Clerk Maxwell and Heinrich Hertz. And it was this basic understanding of electromagnetic waves that enabled the invention, the application of wireless and radio. And so he's really saying that fundamental research is essential. I would say that that's half the story, that you really need both new knowledge and new applications for progress to proceed. Now, this idea that basic information is important and that any application is really founded on many, many discoveries is not a new idea. I'm going to give you a couple of other quotes that I found that I enjoy that bring this same story home. One is by the American author Mark Twain, Samuel Clemens, who in a letter to Helen Keller wrote, "It takes 1,000 men to invent a telegraph or a steam engine or a phonograph or a photograph or a telephone or any other important thing. And the last man gets the credit, and we forget about the others." Another quote, even earlier, comes from 1783, and the American really polymath, Benjamin Franklin, who at the time was the ambassador to France. And he goes out and he sees the one of the flights of the Montgolfier brothers balloons. And someone in the crowd next to him says, "What is the usefulness of a hot air balloon?" At which point Franklin is reported to have turned to the person and said, "What good is a newborn baby?" That there's lots of potential to these things. And to bring this home, I have two more examples. One is a wonderful infographic made by the Quartzoff people. You could go to quartzoff.com and find this infographic because, as you can see, it's very large and impossible to see. But I want to point out what it's really talking about and why I like it so much. First of all, looking at the very top of it, what is this about? It is about all the science that led to the iPhone, to a smartphone, with its Wi-Fi and its touchscreen and its computer and a battery and Wi-Fi going onto the web and its display and the camera and all the things that are in a smartphone. And as you go further down this chart, you see all the science that was needed for these things to happen. Here's a whole set here of different discoveries and inventions that people had to make for each one of those parts of the iPhone. And if you go to the very bottom and look there, now we are more than 2,750 years earlier with the discovery of the first lens and the discovery of binary and working out other aspects, the idea of zero. All of these things, of course, are needed to be able to make that final product. And my final example about this is a wonderful short video that was the winner in 2014 of a competition by the Federation of American Societies of Experimental Biology, FASEB, where they asked people to make a short video to say the importance, to show the importance of basic research. And the people listed at the bottom of this slide were postdocs and graduate students at the University of California in San Francisco. And they made this absolutely wonderful video, which I'll play now and hope that you can hear it. And here we go.
It's a sunny day in San Francisco. We set up a table by the water and asked people this question. If it's the year 1960 and you had $10 to give to science, would you spend it on A, developing an affordable treatment for diabetes, or B, figuring out how bacteria protect themselves? The majority of people we surveyed wanted to spend their money on a diabetes treatment, but what really happened 50 years ago? The government decided to spend money on figuring out how bacteria protect themselves against viruses. The National Science Foundation, the NSF, and the National Institutes of Health, NIH, are two government agencies that allocate tax dollars to scientists. And they decided to fund groups of researchers who thought that studying how microbes protect themselves was an interesting question. At the time, there was no way of knowing how this basic research could affect human health. They were simply curious about understanding something about bacteria biology. So what did the scientists find? Normally, when a virus enters a bacteria, it adds its own DNA to that of the bacteria. In doing so, the virus takes over the bacteria and uses it to make more viruses. Scientists found that bacteria protect themselves from viruses by selectively cutting the viral DNA. This basic finding led to a scientific revolution. Scientists learned how to cut specific chunks of DNA, and they could use this technique to move genes around. They could even move human DNA into bacteria. And because bacteria are very good at making more of themselves, scientists can use them to make large quantities of human proteins from the human gene that they introduced. So why was this technique useful for modern medicine? Well, one breakthrough was the efficient production of human insulin by introducing the human insulin gene into bacteria. This was a very significant advancement in diabetes treatment. Before using bacteria to produce large quantities of human insulin, insulin was expensive, unsafe, and less effective because it was harvested from fetal cows. In fact, the first person to get an injection of cow insulin suffered from a severe allergic reaction. Because of a long line of federally funded basic research, human insulin is now an affordable treatment for diabetes. By asking how bacteria protect themselves, the scientists discovered that bacteria can cut DNA. This discovery was developed into a basic technique used in thousands of labs around the world to create new knowledge and to ask new questions that have led to all kinds of therapies and technology, including insulin, DNA fingerprinting and biofuels, the human genome, growth hormones, cancer drugs, stroke medication, vaccines, HIV meds, antibiotics, and many, many more. Given enough time and funding, who knows what is to come? Many other health benefits and current discoveries would have been nearly impossible without federal funding for all kinds of basic research. Basic research is an investment in our future. It broadens our understanding of the world we live in, and the potential benefits are limitless. Unfortunately, science funding in the US has plateaued, decreasing purchasing power and significantly hurting our competitiveness and innovation. You may not know it, but you and your tax dollars are part of the discovery process. Thanks to you, scientists discovered that bacteria can cut DNA. Thanks to you, insulin, other drugs and diagnostic tools have been developed and are helping patients.
So thank you for supporting basic research.
We are the scientists who depend on you, and we are excited for the future that lies ahead.
Great. So with that, I think there's a question that arises, and that question is, how do we make measurements? How do we make discoveries even more? And I want to make the distinction, or at least give a quote that makes a distinction between making a measurement and making discoveries. And that's by the physicist Enrico Fermi, who said, if the results of your experiments confirm your hypothesis, terrific, you've made a measurement. If the results are contrary to the hypothesis, then you've made a discovery. So the question then arises, how do we make these discoveries? And I want to give you several examples of discoveries that were made that led to Nobel Prizes. So the first example is Robert Wilson and Arno Penzias Physics Prize in 1978 for the discovery of the background radiation in the cosmos. Their experiments started because they were both at Bell Laboratories in New Jersey here in the United States. And Robert Wilson actually was still concerned about the fact that his PhD project didn't work out exactly the way he wanted it to, and had discovered that there was at Bell Labs a previous researcher who had gone and developed and had constructed a 30-foot microwave telescope. And Wilson realized that he could use that to revisit the problem that he had studied for his PhD. And so he and Arno Penzias started working on this 30-foot microwave telescope. Well, the first thing you have to do when you have a piece of equipment is make sure it's working and to calibrate it. So they started it up, and to calibrate it, they pointed it at what they knew was going to be a source of microwave radiation. That was the Milky Way. And they got a nice strong signal. And then to calibrate the machine, they had to have a zero. So they pointed the machine away from the Milky Way into what they thought was empty space. When they did this, it never got to zero. They always had a measurement. They tried everything they could think of to get that background or that recording down to zero. They thought maybe it was nearby electrical lines. Maybe it was airplanes from the local airport that was nearby. Maybe it was pigeons nesting in the machine. They tried everything. They could never get it down to zero. And then they thought about this for a while, and there had been some people that had written some stuff, and they realized that it was not zero for a reason, because they were actually measuring something. What they were measuring was this background radiation in the cosmos, which turned out to be the first real evidence for the Big Bang and led to their Nobel Prize. So you don't even have to do an experiment to get a Nobel Prize. You can simply try to calibrate your machine. Another example like this is John O'Keefe, who shared the Medicine Prize in 2014. He, in his write-up of the Nobel, of his experiments, says that one day he was doing some experiments looking at electrical recordings of cells in the brain, and he was looking for cells in the thalamus when, by mistake, he put the electrode into the wrong area in the brain that he was studying and found that there were some pretty interesting cells there. He studied those cells and realized those cells were responding to the animal's placement in space, and these are called place cells, and it's what he got his Nobel Prize for. Perhaps my favorite of all these stories about these accidental discoveries is Osamu Shimomura's and the discovery of green fluorescent protein, GFP. Shimomura's life is quite interesting. He wrote a biography, a memoir called Luminous Pursuit, which I recommend to people. He grew up in Japan, and at the age of 16, he's told he has to quit high school. He has to go work in a factory. So he goes to the factory. This turns out to be very advantageous because the city that he grew up in was Nagasaki, Japan, and by working in the factory, he was outside of the city when the atomic bomb destroyed the city. He writes about walking the 10 miles home, and by that time, the radioactive ash had turned his white shirt to a sort of ash gray, and he attributes his grandmother's insistence that he take a bath and get out of these clothes that probably saved him from radiation poisoning. But there was no school to go to after the destruction of the city, no college. But he eventually went to the first college they built there, which was the College of Pharmacy. He says he was not that interested in pharmacy, but he was interested in going to college. Afterwards, he goes and works as a technician in a lab and develops his lifelong love of a really fascinating biological problem. How is it that different organisms can actually generate light? And we know that many different organisms do so: fireflies, glowworms, jellyfish, bacteria, all sorts of different organisms can generate light, and they do so chemically in very different ways. And so that became his interest. He started working on this, made a very interesting discovery as a technician, and on the basis of that was invited to come to the United States to do further research. And his advisor at Nagoya University, where he was working as a technician, realized that if he came to the United States not as simply a college graduate, but as someone with a PhD, he would be paid twice as much. So his advisor arranged to give him a rather unusual going away present, his PhD, which he had. Goes to the United States, and one of the things he does is try to understand how this jellyfish, Aquaria victoria, produces light. He spends an entire summer trying to isolate the protein that's needed to generate light in this organism and fails completely. Nothing works. He's a very accomplished biochemist, but absolutely nothing works. He cannot isolate the components from the jellyfish. One night, after another failed experiment, and it's already gotten to be dark outside, he decides to give up and go home to have dinner. But before he does, he decides to clean up. And he takes all the samples that he had and he throws them in the sink, which had some seawater, probably some other, maybe jellyfish parts or whatever. But he throws the stuff away in the sink, turns off the light, and is about to leave when he looks back at the sink and discovers it's glowing in the dark. He's a little surprised at this, but then thinks about it a little bit and realizes that maybe there was something in that seawater that he never had in his preparations. He goes back to the lab eventually and does the experiment again, but now adds something that he knew was in seawater, but had never been in his experiment, and that was calcium ions. And when he adds calcium ions to his solutions, he gets a flash of light. He uses that to purify the protein, which he names after the jellyfish. He names it aquarin. And aquarin plus calcium ions produce this beautiful light. So a completely accidental discovery that he follows up and is able to answer his question. What is the protein that makes light? And it's this protein, aquarin. But then he has a problem. It's the wrong color light. It's blue light. The jellyfish produces green light, and he can add calcium to all his other samples. He never gets blue light. So he thinks about this for a while and says, maybe there's something that converts the blue light to green. And so he shines blue light onto all the other samples. And sure enough, a completely different sample does have a protein in it that when you shine blue light on it, you get green. He uses that to purify that protein. Actually, in his write-up of the purification of aquarin in 1962, he has a footnote that says there's this other protein. If you shine blue light on it, it will give off green light. And he called it the green protein, but we call it now the green fluorescent protein. And when GFP is present, you can actually get green light. So a second accidental discovery because he got the wrong answer. It was blue light, not green. And this wonderful molecule is really quite interesting. It is a fluorescent molecule. You shine blue light on it. You don't need calcium. You don't need anything else. You just need blue light. It will give off green light. And that's rather remarkable. The molecule itself is beautiful, as you can see in this diagram. It really is one of the few molecules in the world that I believe looks exactly like you would expect it to look like. It looks exactly like the lantern it is supposed to be. So GFP looked like a really interesting discovery, but no one really worked on it for many years. I heard about it in 1989, and in collaboration with a friend, we started looking at it, and you can use GFP to look at genes and proteins. Now, normally a gene has a promoter element, this little arrow at the beginning that basically is the instruction of where, when, and how much of a product can be made. And then you have the coding portion of the gene, which I've diagrammed here as these 3 exons. And the idea is that if you add now GFP, you can do some very interesting things. You can replace the protein with the sequence for GFP, and this makes what's called a transcriptional reporter. And it will tell you where, when, and how much a gene is turned on simply by looking at its expression. And this is exactly what a graduate student in the lab, Gia Skirkin, and this was all in collaboration with the person who identified the coding sequence for GFP, Douglas Brasher. What they did is what Gia did is she put it into bacteria. All of a sudden we had green glowing bacteria. And then later in the lab, we put it into C. elegans, the nematode that we study in the lab, and you can see the image of a set of nerve cells where only those nerve cells are lit up because they're the only cells that express GFP. But in addition to making a transcriptional reporter, which will tell you where and when a gene is turned on, you can make a different construct, a construct in which the GFP coding sequence is added to the normal protein that's made. And this is now called a translational reporter. And that translational reporter, the first one that was not made in worms. It was made in fruit flies by Tula Hazelrig, and she was able to show that a protein made in those cells on the left of this image, which are the cells called nurse cells, make a protein that then is transported into the cell on the right, which is the growing oocyte in fruit flies. This is keeping everything in the family because Tula Hazelrig is my wife. And so it was really one of the first people to realize the importance of using GFP in experiments. So I'm going to show you a couple of movies with GFP to let you see something about showing life happen. The movie on the left has GFP expressed in a protein that's part of the spindle of cells dividing. And you're going to see sped up 4 rounds of cell division here. These movies were taken by a graduate student, I've never met her actually, named Rosalind Silverman Gavrilla, who put them on a website for the American Society of Cell Biology. And as you look at this movie, you'll see that all of the divisions appear to take place at the same time. In fact, she named this movie In Synchrony. And being able to watch this happen really invites a number of interesting new questions. What is coordinating these cell divisions, these nuclear divisions? How can these be working? And people have studied this aspect of cell division quite a lot. Now, on the right-hand side is a colored image, but it is a falsely colored image. And the convention is that the less you have of something, the more you're towards the blue end of the rainbow. And the more you have, you're towards the red end of the rainbow. And so this is a falsely colored image, but again, using GFP. But now it's a different GFP. It's a GFP that has attached to it a small peptide that will bring it into the nucleus. And we're going to watch 4 other divisions, so a similar set of images as the one on the left, different animal, but again, early fruit fly. And when the nuclei are intact, this material is just in the nuclei, as you can see in the still image now. But when the nuclei break down for cell division, then it's going to be all over the early embryo. So let's watch that movie. She calls this Starry Night after the Van Gogh painting, and you'll see why it looks very much like that. The nuclei now have absorbed all the GFP. They then break down, and it's all over the cell. But the interesting thing about this movie, in comparison to the one on the left, is that things seem to occur not simultaneously, but going from the lower right to the upper left-hand corner. So one could ask, why is this different? And part of it may be that this is a slightly damaged preparation, that in making the slide, her thumb may have been a little heavy on the slide. At least that's one of the suggestions that a friend of mine made, having done similar experiments. So this allows one to look at things, but people are never really satisfied. There's always new things, but once you've made a tool, you can change it. And one of the really interesting series of changes was started by Roger Chen, who also shared the Nobel Prize with us, with Shimobu and myself in 2008. He modified GFP so that it made a blue fluorescent protein called BFP and a yellow-green protein, which was called YFP, or sorry, yellow fluorescent protein, also called citrine. That was about the extent of how GFP could be modified to produce different output colors. But then a wonderful discovery was made by Mikhail Matz and Sergei Lukyanov, looking at coral, which they knew was fluorescent, and they were able to find the first red fluorescent protein called DS-red. This then led to other people going off and looking for other fluorescent proteins, but it also led Roger and others to modify the red fluorescent proteins to produce other colors as well. And you get now fluorescent proteins that span the entire visible spectrum, and actually even some now beyond it. And if you go to this website, which is fluorescentproteinbase.org or fpbase.org, there are now over 1,000 fluorescent proteins that people have either generated or have found in the wild that people can use for various studies with a lot of very interesting properties. But what does color give you? It gives you a number of things. One of them is that you can start to look at multiple things at the same time. And Jeff Lichtman and Josh Saines at Harvard developed this very nicely to label different nerve cells in the mouse brain in something they referred to as brainbow. And here are some of their images. And it's now been used in fruit flies and zebrafish and other systems to be able to label cells. There's been other multicolored systems that have been developed, one in C. elegans, for example. But another experiment that I quite liked that used different colors was an experiment by Jeremy Nathans at Johns Hopkins. He was interested in looking at something that occurs in females in female mammals, which is called exon activation. that while they do have two X chromosomes, one of them is inactivated in every cell. And he wanted to know, is that really true? Is one X always inactive in every cell? And to do that, he made mice that had on one of their X chromosomes a gene that expressed red fluorescent protein, and in another one on the other chromosome, the same gene, but now expressing green fluorescent protein. So red and green. And here are some of his images, and you can see there's no mixing. There's one or the other in these, and he looked at many other tissues as well. There have also been some very inventive uses of GFP. And I think this is one of the important aspects about useful or useless information. And that is, once a discovery is made, it enables other discoveries and other uses, things that the original developers never had in mind. I like to think about this in terms of the laser. When Charles Towns developed the maser here at Columbia, and then that led to the development of the laser, those experiments were really experiments in basic physics. They were not thinking, no one had come to them from the grocery store and said, you know, I'd really like to be able to help the checkout people get people through the lines faster. We need something to read codes on produce. And nobody from the record or film industry came to them and said, we'd like to have CDs or DVDs as ways of recording information. And no surgeons came to them and said, we need to have something to do better eye surgery and other types of surgery. But all of those applications did come after the development of the laser, as well as other fundamental science discoveries, low temperature physics, laser tweezers, gravity waves, all of which were enabled. These are all things that won Nobel Prizes, were enabled because of the initial discovery and development of lasers. So two inventive uses of GFP include the work of Jeff Waldo, who was interested in looking at protein folding. And he made the very nice observation that if he made a protein fusion of his protein and GFP, if his protein didn't fold, GFP would not fold. And if GFP doesn't fold, it's not fluorescent. So you can shine blue light on it all you want, you don't get anything out of it. But if he then mutates his protein so that it folds correctly, then GFP will fold correctly. And now when you shine blue light on it, you'll see green. So if you look at proteins being made in E. coli, the ones that are folded correctly will be able to be fluorescent. You'll be able to pick out those colonies because they're green under blue light. Another example is work by Lynn Regan, who was at Yale as a chair of the department, and she did a very interesting chemistry department, did a very interesting experiment. The experiment was cut GFP in half. So she cut GFP in half, and as you might imagine, cutting the molecule in half basically means it doesn't work. But then she did something very clever. She added to each part of the GFP, she added a domain from what was called a zinc finger. Now, the two domains of a zinc finger, or leucine zipper rather, can come together. And when they come together, the two split parts of GFP can come together. And when they do, they become fluorescent. And what this does is it gives a model for how we can look at protein-protein interactions. How if you put your two proteins that you think might interact together, if they come together, then the two parts of GFP can come together and you can actually find that there are the protein interactions. I think both of these are wonderfully clever consequences of being able to have fluorescent proteins. But there have been many other uses, and some quite unusual, and to go through them very quickly. Researchers in Japan made fluorescent silk, and they made fluorescent clothing out of that, because the silkworms could make silk with GFP or RFP and other colors added onto them. Other researchers made cellular lasers. Bob Burlage used it as a detector for the TNT that leaks from landmines. The artist Eduardo Carr arranged for a biotech company to make a green fluorescent bunny that was a family pet, that he used it to encourage people to think about the connection between science and art and science and technology in his various art shows. And finally, GFP has actually been the basis of maybe the first use of GFP in a human is this example, which comes from the movie maker Ang Lee, who used GFP as the reason in his movie Hulk to explain why the Hulk skin turns green. It actually starts off with getting an extract from a jellyfish, which is quite wonderful. I actually know the screenwriter, James Shamus, for this movie, and he's here at Columbia, and I went to him and I said, James, this is wonderful. How did you know about my research? And he said, I have no idea what you work on. It turned out that there was a student from MIT who is working on the film set and convinced them that this would be a great idea, a great explanation. I don't care who he got the idea from. I think it's quite wonderful that the reason the Hulk turns green is because of GFP. In any case, you can find other examples of the use of GFP. Mark Zimmer at Connecticut College has a wonderful website. If you just look up Mark Zimmer and GFP, you'll be able to see other examples. But again, to go back to my question, if many or most discoveries are accidental, how can we actually increase serendipity? How can we promote this? And I think there's two basic ways. The first is, I think we have to start questioning more things that are right in front of us, that are around us in our world. And the other thing is to question our assumptions. So let's go to the first thing. My first scientific paper came about because I was studying a major transparent part of the body. Now, I can't do this online, but when I get to this point in this slide to an audience, I usually ask them, how many of you know the transparent part of the body? And I can tell you that about 5% or less of the audience raises their hand. And they really are sort of mystified. So I give them a hint. The hint is, it's right in front of your eyes, because what I'm talking about is the cornea. Now, the interesting thing about this is something we have used essentially all our lives. But I have found very few people who have ever realized that this is an interesting scientific problem. How can one make a tissue that is transparent, that allows us to see? And even less that have actually asked the question about it. And I was very fortunate. I had not thought of this question, but I went to a lab where people were thinking about this. And I find it sort of fascinating. I'm going to give some other examples of things right around us. And for this, I'm going to tell you about my second cousin, Ray Goldstein. Ray is a professor of physics at Cambridge University in England. And he's made a career I think, of being able to find interesting problems that are all around us and asking some very profound questions about them. In fact, one of the first things he did was ask the question, what happens when a bunch of fibers are all together? How do they move together? But to think about that and to find a aspect of the real world that this occurs, he asked the question, what determines the shape of a ponytail? And he worked out how ponytails have the shape that they do. And this earned him the 2012 Ig Nobel Prize in physics for figuring out how a ponytail shapes. But he's also written wonderful papers on why icicles have the shape that they do, why our clothes don't fall apart since they're made of really tiny fibers. And finally, and most recently, a year or so ago, he did a paper on why, when cicadas emerge from the ground, these insects do so as swarms. It's not how do they know what year it is to come out, but why do they all come out together as a group? And he and his wife, who's a mathematician, worked out the sort of aspects of this work that would enable them to say there must be some sort of communication among these buried insects before they emerge. Another person who I think did a spectacular experiment of something right around her is I met her as a high school student, Abigail Freed. And I every year or many years, I go to the International Science and Engineering Fair, the ISEF, which is a culminating worldwide science fair that has people from all over the world, about 2,000 people that have won local science fairs in their countries and then come to compete. And Abigail had come and she had her display of her project. And I happened to be walking by and I said to her, what is your project about? And she said something to me that you don't often hear. Maybe no one else has ever heard this as the beginning of a scientific talk. Her opening line was, well, the thing you should know about me is I am a ballerina. And I'm actually a very good ballerina, almost at a professional level. That's rather unusual as a start for a scientific talk. And so I was intrigued. And she said, there's a problem. It's the problem with the shoes. Ballet dancers use shoes that have a cardboard sole to them, and the shoes lose their flexibility after only one performance. So they have to buy a new pair of shoes for every single performance. That's very expensive. But they need that flexibility in the shoe. And so she decided that was going to be her project. And so she found a carbon composite material and figured out the right flexibility of it. Here's a picture of her showing the various flexibility things. And the middle one, it turned out to be perfect. And that was her project. She had found a better sole for ballet shoes, something that would last literally for months and would allow the shoes to be used repeatedly. I thought this was terrific. Again, a problem right in front of things. So the other aspect of this is the idea of questioning assumptions, not only looking at what is around us all over and asking, why is this happening? But asking, what are our assumptions? So I want to talk about that a little bit. Now, when I was younger, and I must admit, still now, I'm intrigued by what are called matchstick puzzles. I think these are good examples of thinking about assumptions. When you first start out looking at matchstick puzzles, and there's websites you can go and look at all of these things, they will give you a question like this one. Move 3 matches and get 3 perfect squares. And you move things around, you fiddle with this, and you eventually find that there is an answer, and that's one possible answer here. So that's very nice. And then you get another puzzle. The next puzzle says, move 3 meshes and move the fish around. And again, you move things and you suddenly realize that this is a pretty good answer for that question. You can get the fish to move in the other direction by just moving 3 matches. Well, these are all great until you get to the next puzzle. The next puzzle is make 4 equilateral triangles from 6 matches. And you can move these around the plane a lot and get very frustrated until you suddenly realize that maybe your assumption is incorrect. You've been thinking of moving things only in 2 dimensions. If you don't think that, you say, well, what if I can move them in 3 dimensions? You get this figure, which will have 4 equilateral triangles, but being made as a pyramid. But that's actually not the only answer, because there's another assumption that you're making. And that assumption is, why are all the triangles the same size? If they're not the same size, this is another possibility. Excuse me, with 2 small triangles and 2 large triangles. So after you've gotten over those assumptions, you get another problem. And that problem is, make these 3 matches into 6. You cannot break them in half because half of them will not be matches, so that's not going to work. So how do you make them into 6? And you realize maybe that, again, it's your assumptions that have been holding you back, that you've been thinking about shapes. If you think about something other than a shape, you can change it to 6 by just moving the matches to make Roman numeral 6 rather than Roman numeral 3. Now, you might think about these as just exercises and expanding your thoughts, but I want to give you an example of someone who actually uses this idea of questioning assumptions. And that's my friend Mu-Ming Pu, who's a neuroscientist. He was at Columbia. He then went to Berkeley. He's also been the head of the Neuroscience Institute in Shanghai. He's a spectacular scientist. And one day, we were all sitting around, and I don't know if I asked him or somebody else asked Mu-Ming, where do you get your great ideas from? You get so many wonderful ideas. Where do they come from? And he said, well, I use what I call the retrospective approach. What is the retrospective approach? He finds an interesting hypothesis in a textbook. And for him, it was the 2nd edition of the textbook, The Molecular Biology of the Cell. And once he finds that interesting idea, he says, why do people believe that? And so he'd go back into the literature and he'd find out what was the basis of that idea. And usually it was an extrapolation of an experiment that they weren't really able to get a definite answer for. And so he would then retest the hypothesis using more modern methods. And he would either find that he confirmed the result, and you have a very nice paper because he would be able to confirm and give support to an idea that other people had had. But many times he found that they were incorrect. And so he could then publish the correction and then go back to the book and find out what other hypothesis was really not based on as good a data as it should have been. And so he's done this in his career, and it's a very good way of starting, just asking, why do I believe this particular piece of information? I want to end by giving 3 favorite quotes of mine, or at least 2 favorite quotes, and then one I'm going to let Flexner have the last word in all of this. So the first quote comes from Robert R. Wilson, who was the first head of the Fermi Lab, the precursor or the world premier particle accelerator before the CERN super collider. And when the Fermi Lab was, before it was made, there was a big debate in the US Congress about whether they should actually fund this. And Wilson was asked to come and talk to members of Congress. And they asked him a bunch of questions that basically were, okay, tell us what, because this was during the time of the war in Vietnam. There was a lot of concerns about security and national defense. They said, tell us, how many ways will this accelerator help national security? And he gave them the answer they did not want to hear: none. So they asked this again and again. Particularly Senator John Pastore of Rhode Island was the person who asked questions. And then finally said, no, look, Dr. Wilson, will you please tell the members of the committee, in what respect will this accelerator help the national defense? At which point Wilson gave the following answer. He said, it has only to do with the respect with which we regard one another, the dignity of men, our love of culture. It has to do with, are we good painters, good sculptors, great poets? I mean all the things we really venerate in our country and are patriotic about. It has nothing to do directly with defending the country, except to make it worth defending. My second quote comes from somebody very close to me, my daughter, Sarah. And she said this in 2004, so quite a while ago. At the time, she was 12 years old, and I had been elected to the National Academy of Science. But I think what she said is very important because I think it's one of the real joys about science. The induction ceremony to the National Academy of Science is perhaps one of the most boring ceremonies in the universe, because everybody is there to see the one person that's being inducted, but there's about 100 people that are being inducted. So you have to sit through all these other people. And so what does each person do? The person gets up on the stage, shakes hands with the president of the National Academy of Sciences, then has their picture taken with the president of the National Academy of Sciences. Then they walk across the stage. There's the book. They tell you that every member of the National Academy of Sciences has signed the book. And so you think you're signing the book that Einstein signed. But then they tell you it actually started in 1970s. It's not as exciting, but you still sign your name in the book, and that's quite nice. And while you're walking across the stage to sign the book, and while you're signing, someone reads a sentence or two about why you were elected, what you did to get yourself elected to the National Academy of Sciences. And I knew, as I was going through this, that my daughter probably didn't have all the patience in the world to listen through all of this, or at least that's what I thought. And so when I made my way down to where my wife and daughter were seated, I went to my daughter and I said, you know, Sarah, I'm really happy that you're here to help me celebrate, but you must be bored out of your mind. I'm so sorry that you've had to sit through all of this. And she looked at me as she has often looked at me throughout her life and said, Dad, don't you understand about these people? And I went along. I said, because she had been listening to all those sentences. I said, what do you mean? And she said, they were the first people in the world to see or do what they did. The first person in the world. That's what is the joy of doing scientific research. I'm exceptionally proud of this answer that she gave and this insight that she had. But I'm a proud father regardless. But it really is one of the real joys that we have, that we get to find out something first of everyone in the world. And that's, I think, very exciting. I want to end with another quote from Abraham Flexner, because I think it's especially important these days, but it also comes from his 1939 article. In it, he says that is fundamental, of fundamental importance to have absolutely untrammeled academic freedom. And he said in 1939, in certain large areas, Germany and Italy especially, the effort is now being made to clamp down the freedom of the human spirit. Universities have been so reorganized that they have become tools of those who believe in a special political, economic, or racial creed. The real enemy of the human race is not the fearless and irresponsible thinker, be he right or wrong. The real enemy is the man who tries to mold the human spirit so that it will not dare to spread its wings, as its wings were once spread in Italy and Germany, as well as in Great Britain and the United States. Thank you all very much. I'd be happy to take questions.
Thank you so much for this beautiful, amazing talk. Very inspiring. I'm gonna read some of the attendees' questions and then I'll give you the chance to answer. First question is from Christy Ad, chemistry student here at AUB. Her question, throughout your career, have you ever had to reframe your scientific intuition after being proven wrong by unexpected data? How did that moment reshape the way you approached uncertainty and risk in future experiments?
So I have had quite a lot of truly wonderful ideas that have not proven out in any way. And so you just keep going. If you have 2 ideas out of 100 that work, you are spectacular. You're doing a great job. I think that we always look at things, it's really easy to fall in love with your ideas, but when something comes up that says that's the wrong way, you have to think of something else and you have to try something else. So we're always pivoting from one brilliant idea that turned out not to be so brilliant to another. So I think it's just a part of being a scientist. I want to add one more thing. I want to talk about that quote from my daughter about being the first person in the world to make a discovery. I think that by its very nature defines that we are going to fail a lot of times. If we're trying to be the first person in the world to discover things, we're going to make a tremendous amount of mistakes. It's inevitable. We don't know what the right answer is. We're trying to find that out. And it's the trying that makes us get an inch one way or another along the path of discovery. So I think that by its very nature, it's not fun to fail. It's not fun not to get the right answer, but it's, I think, inevitable because of what we're trying to do, which is gain real understanding.
Thank you. This question is from Taleen Sariyuddin, chemical engineering student. You mentioned in one of your interviews that even after becoming a Nobel laureate, many of your papers still got rejected. What was the driving force that kept you motivated and determined to proceed in your research, even though you knew that your discoveries might not be easily accepted?
So I don't know if it's so much easily discovered. There's an interesting thing. If once you get, this tap on the shoulder that you've gotten a Nobel, it doesn't mean you're a great scientist. It doesn't mean that everything you do is going to turn to gold. And you shouldn't be judged any differently than any other scientist. And so, my papers were not always accepted initially before I got the Nobel, and it didn't happen after I got the Nobel. I was the same scientist. You don't get a free pass. You shouldn't get a free pass simply because somebody has given you an award. And I want to say something a little bit about the Nobels, at least how I think about them. It's not a prize, I believe, for the smartest scientist, for the most accomplished scientist, for the scientist with the most funding or the biggest lab or the most papers. It is about something that you cannot really go after, or very few people go after. And that is, it is a prize given to science that has changed the way we think about science or that changed the way we do science. So it's not that we showed this great result about GFP. It's the fact that so many people then found it very useful to use and that it really had a life of its own. They gave the prize to GFP, not to the three of us that happened to be people involved at the beginning. I think that for many of the prizes, I did my postdoc with Sidney Brenner, who was a Nobel laureate in 2002. Sidney used to talk about three types of ways of doing science. One way was to be the person that finished the story, that gave the final touch. Now nobody had to do that anymore. He said that was exceptionally hard. He preferred the other way. The other way was to start something new. And he was able to do that in many ways. And he was an absolutely brilliant scientist. He would then say that all the rest is in the middle, but it's in the middle that all those interesting discoveries come about. These new things that are unexpected, that lead you in different ways. But it's the impact of how the discovery was used by people. Shimomura discovered GFP in 1962, but it took all that time till 2008 for him to be recognized for the really wonderful discovery that it was, not because discovering a fluorescent protein was important, but because there was this sudden realization of how useful and what an impact that discovery has had on science. And, they could have just given the entire prize to him. It was really a wonderful, wonderful discovery.
Thank you. This question is from Carol Castoon. On the Struggling Scientist podcast, you describe music as a joy of patterns and discovery. Do you see a link between guitar improvisation and experimental design? Has music shaped your approach to science?
It has not really affected my science. For those that are not familiar with this, my father was a professional guitarist. And since I was the oldest of his three sons, when I was about 12, he gave me a guitar and I've been playing ever since. So I've been playing for a long time. I'm still trying to master the instrument. It's going to take more than a lifetime to do that. But I enjoy music a lot. I have enjoyed trying to compose things. It's very difficult to me, very hard for me to do that, but I get a lot of joy from it. I love playing pieces and enjoy the physicality of being able to produce a sound and enjoy the music and enjoy what other composers have written. I don't think it's really affected my science. It's affected my life greatly because it gives me a real joy to be able to do that. So I don't think being a musician is necessary to be a scientist. I just find that it has been a wonderful part of my life.
Thank you. Can you stay 5 more minutes just to answer 3, 4 more
Absolutely.
questions? Thank you so much. This question is from Ghadi Sukkariye, a chemistry student. Looking back at your journey, what failure was the one that pushed you to success the most? And what do you think will be different today if you had not experienced it?
So let me tell you the biggest failure of my scientific career. I was a junior, or just finished my junior year in college, and I went to a lab because I was a biochemistry major. I knew it would be a good idea to work over the summer in a lab, so I went to work in the lab. And I applied a bit late in the year to get into the lab, but I was admitted to a nice lab, but they had no room. But there was an empty students' lab that was 50 meters away. And so I set up to do my experiments in this completely empty room. I also had perhaps one of the dumbest ideas anyone could have about scientists. And that was that scientists had to had to do it on their own. If you didn't do it on your own, you were not really a scientist. So to prove yourself, you had to come up with things on your own. I failed completely. I couldn't do the experiment. It was the worst summer of my life. And so what did I do? I quit. I said, that's it. I have now proven to myself I am not a scientist. And so I finished my senior year of college by taking a whole bunch of different courses. I had a wonderful time learning about all sorts of different areas. And then I graduated. And I went and did a whole bunch of short-term jobs. And then I got a job teaching high school. And for 2 years, I taught high school. In the middle of that time, in the summer, between the 2 years of high school that I taught, I made a discovery. And that discovery was high school students have the summer off, but high school teachers have to find a job. So I needed to find a job. And what I did, I talked to a friend. We were near Yale University, and she said, well, I know somebody who might hire you for the summer. And I was hired into the lab where they were studying what makes a cornea clear. And I went in for the interview. I wasn't really sure I wanted to be a scientist again, but this was a summer job. This was fine. And I went in for the interview, and I was told about the work. And it reminded me of a paper I had written about, a short essay in college several years earlier. And I completely misremembered that paper, except for the conclusion and one other aspect about it. That paper and this new lab used the same apparatus to study the flow of ions across a membrane, in their case, a cornea from frog. And they were studying chloride ions. The paper I remembered was not frog, it was toad, not cornea, but skin, and not chloride, but sodium. It was completely different. But they used the same apparatus, It would be like saying to somebody, oh, you work with you work with gels. I once read a paper about gels. Do you get the same answer? But I asked the question anyway. I said, does cyclic AMP, which the first paper had, does that work in this system? And the man I was applying to, scientist, I think he was so surprised that I asked a question that he said, I don't know, but you're the second person in 2 days to ask me that question. And I think because I asked a question, as misguided as it was, he hired me, which I consider the first miracle because I had really no skills at all. And he hired me. And then the second miracle occurred. The second miracle was that 1 week after I started in the lab and was starting to learn the procedures, he walks into the lab and tells everybody, this is the summer of 1970, he says, goodbye, I'm going to Europe to do some work on my own. Have a good time. And he leaves the lab. So he's gone. And he gave me a project that was actually pretty boring. I won't go into what that project was, but he was gone. And, this 1970, there's no emails, there's no faxes, there's no way of, and he didn't want to communicate. He didn't say, send me a letter or anything. He just left. So I found the other person that asked that question. And together I asked him, I said, well, how would you test if cyclic AMP is working? And he said, well, I would put adrenaline on it and see what happens. So I did that. And it went off scale. It worked beautifully. And I learned the difference between the first time when I was in college and this time was I had people that could help me put things together and direct me to where to go, and I could ask advice and support, and I got it, and it was wonderful. And the experiments worked. That was the other nice thing that happened. And I spent the whole summer on my experiment. And the one thing I could do is I could go to the library and look up things. And so I found all sorts of literature that was germane to this issue. And so when the guy came back at the end of the summer, he said, oh, so tell me about the experiment I asked you to do. And I had to say, I didn't do that experiment. But here's what I did do. And I showed him all the results. And he was very happy. And he did another experiment. And together we published what turned out to be my first paper. So I'm not saying this as what you should do, but that's my story. I started off and I had completely failed. I completely had no confidence in myself at all. And then I had the wonderful opportunity to go back and try again. And I realized that I could come up with some crazy ideas, maybe not really well-founded, but at least I could come up with some ideas. And every once in a while, an experiment worked. And that was pretty exciting. And with his support, I applied for graduate school and then went on to graduate school, where I had another exceptionally supportive advisor, which I think was very good because I still didn't have a great deal of confidence in myself. But, you muddle through.
Thank you. One last question from the
Sure.
audience. This is from Lara Mirai, environmental health student. How do you navigate differences in perspectives, personalities, or disciplines within collaborative teams?
That's a great question. It's very hard. I think everybody needs to be looked at as an individual. And sometimes that's easy. Sometimes it's not as easy. I don't know if I succeed every time. I don't think I do all the time. But everybody comes with their own history and their own perspectives. And I find that the easiest collaborations I have had, and mainly these have been with graduate students, is I have had a wonderful time with many graduate students who come to me and say, I have an idea, or I tried something, and this is what I have, and I want to talk to you about it. And we'll talk about it. And it's not so much what they did, but in the course of it, I'll either say, wow, that's great. What are you going to do next? And they'll tell me, and they go off to do what they're going to do next. But every once in a while, I might say, I wonder if we need another control, if we need something else, because here's an alternative hypothesis that might be possible. And they will discuss it. Discussing it means is another word for argue. What it'll be. And we'll argue and we'll discuss the idea. And sometimes they'll convince me, yeah, maybe I shouldn't have brought that up. Maybe they've been able to counter that. And every once in a while, they will say, yeah, maybe you're right. And then what they do is something very interesting. They say, yeah, I'll go look at that. And they go off. Their ego hasn't been upset by the discussion. My ego hasn't been upset when it goes the other way. I'm just delighted of what happens. Their ego was not involved. That's sometimes very difficult for people to give up, that we are actually a team, that we're actually working together. And when that happens, it's really spectacular because you get a lot more done. And it's very exciting because it's not one person thinking about the problem, it's more than one. And to me, that always improves things. So I'll go with that.
Thank you very much. Dr. Toby, do you have a question or comment? I don't know if when he was referring to the rejection of his papers, they were with Angewandte, but you can answer that right now.
I do not have a question. I think it's been a wonderful presentation, and given your last answer, there's no better way to end it with the idea of teamwork and collaboration. So
Absolutely.
I think it's best left at that. Thank you very much.
Thank you.
Thank you.
It's been fun doing this.
I want to thank you on behalf of all the attendees for this beautiful, inspiring talk. I'm sure everybody was enjoying it and inspired in his or her own way. I hope I will have the chance to host you here in person in our beautiful campus, AUB. But I will be writing to you again, hoping I can convince you to come. We have a beautiful symposium organized in September to celebrate the 160th anniversary of AUB. And it will also be organized in partnership with Angewandte. And Dr. Toby promised me that she and Dr. Maz will come to Lebanon, and that will be their first trip. We will have for the first time the editor-in-chief and the executive editor of Angewandte. Well, you said you will try your best, right? So your best is good enough for me.
We did.
Susan,
We
yeah.
did, indeed.
Great.
Hopefully, I will be able to convince Professor Chalfie to come. It's a beautiful campus, and our symposia are always very special. Thank you again for your time, and I wish you a great rest of the day. It's
Great. And good
almost
evening to
noon
you
right
and
now in
everybody
Colombia.
else listening there. Thank you. Take care. Goodbye.
Thank you. Have a great evening, everybody. Thank you. Bye-bye.
Hey.